Hello, everybody, and welcome back to this webinar series in biotechnology and agriculture. This webinar series are being developed with the movement of solidarity for Africa. And this webinars were developed jointly with our strategic partners, OCP-Africa and RUFORUM. Okay, today I have a great pleasure to host Professor Fawzi Bekkaoui, who is the director of the Institut National de la Recherche Agronomique, INRA, in Morocco, which is the National Institute for Agriculture Research in Morocco. Professor Fawzi will elaborate on one of the very hot topics, which is disruptive technologies in agriculture. So again, welcome back to this webinar. Thank you for your participation and for being always present to this webinar series. I would like also to thank our partners, OCP-Africa and RUFORUM, for putting this in place for you. As today we have Professor Fawzi from INRA. Before he starts his presentation in his webinar, I would like to ask him a few questions about INRA. So, Professor Fawzi, so could you tell us a little bit what is INRA, what's its mission, and what kind of research activity?
First of all, I would like to thank you, Professor Aziz Yassri, for this invitation, kind invitation, and thank UN6P, ESAFE, OCP-Africa, and RUFORUM for giving me this opportunity to share some ideas about agriculture and research in agriculture. I am with INRA for the last year and a half. INRA is the research institution in Morocco that's focused on conducting research for the benefit of the Moroccan farmers. We do research in various areas of agriculture, including the development of crop varieties, whether they are cereals, legumes, or other trees like olives, citrus, palm trees, cactus, argan, for example. We also have a responsibility of conservation of natural resources, in particular crops and plant species. INRA has a gen bank that collects some of these races or lines in Stade, Morocco. We also do research on soil for the conservation, preservation of soils. Do also some research in animals, in particular sheep and goats. Those are some of the essential activities. We are an institution of about 600 people located in 10 regions of Morocco. And we have also 23 research stations where we do most of our trials. I can't hear you, Professor Aziz.
My microphone was muted, I'm sorry. Thank you for this overview about INRA and its mission and the research activities that you are undertaking now. What is today, in terms of high priority research for INRA in agriculture domain?
So we go through 4-year cycles of programmation. We are developing the next program starting in January. Now you can maybe, because there's the feedback. So yes, we are in the 4-year cycle. We are going to start a new program in 2021, January 2021. So Morocco had, in the last 10 years, had a research strategy. He had a program, a plan called the Plan Maroc Vert, Moroccan Green Plan, that's focused on developing agriculture for the benefit of the agricultural industry. This plan has ended, and we are starting a new plan called Generation Green. And this plan will focus on various aspects. First of all, on the social aspect, whereby we will try to improve the revenues and the social status of farmers. There are a lot of small farmers in Morocco that have relatively small revenues. And through the improvement of agriculture, we would like to improve the revenues of these farmers, moving into a middle class category. And there are various aspects to that. And on the other hand, we are going also to implement and develop new technologies that are going to help the Moroccan agriculture. For instance, implementing more tools in digital agriculture, implementing and introducing more practices in organic agriculture that's present in Morocco, but not at very high scale, and there are a lot of opportunities in these areas. And also focusing on soil improving and preserving the soil. Those are just examples. This plan is a very ambitious 10-year plan that has many objectives, and I invite those who are interested to consult the website of the Ministry of Agriculture of Morocco.
Thank you, Fawzi. So now, how about the kind of relationship that INRA is establishing with the public organization, research or academic in Morocco outside?
We have many partnerships. The first one is within the Ministry of Agriculture. We have a consortium that's has as partner Institut Agronomique Hassan II, École Nationale de Meknès, École Nationale d'Agriculture de Meknès, l'INFI, l'École Nationale de Foresterie de Forêt à Salé. Plus en dehors du ministère, au niveau de la recherche, on a des partenariats avec plusieurs universités, notamment l'Université de Rabat, l'UM6P, l'University of Mohammed VI Polytechnique, University of Marrakesh, and other universities in Meknès, Kenitra, and Nimlel. So there are others, and some of these have been ongoing for a while because we host a lot of students, PhD students. We have more than 100 PhD students, and these students are registered, of course, at this university. INRA does not deliver a degree, but we host students at the level of the PhD and the master level. Of course, we have also international collaborations, and that include INRA France. We are discussing with other partners in Spain, in Australia. A strategic partnership we have is with ICARDA. ICARDA is one of the CGR centers. And ICARDA, one of its major centers, is located in Rabat, and we have very close relationship with ICARDA through various projects. We do some of the research trials, and we provide land and space to ICARDA, and it's very, very important collaboration for us. So those are some examples, and I'm sure there are others. I didn't. This is on the academic research, but the other partnerships that are key is with industry and with the associations. Through the Morocco Green Plan, there's been more than 18 associations representing the farmers that have been created. And these associations that are at the early stage will play a very important role in research, development, marketing, representing the farmer, their needs, and also how to enhance their revenue and how to enhance their products. And so we have established partnership with these who fund some of the research and help us also identify some of the priorities of research. So we value this partnership, and we are going to continue working with these and enhance them in the future.
So thank you, Fawzi. Again, if you have just joined us, So today I'm hosting Professor Fawzi from the Institut National de la Recherche Agronomique. So before I hand it to Professor Bekkaoui for his webinar, I would like to introduce him to you. So Dr. Fawzi Bekkaoui is director of INRA Morocco. He was previously director of the School of Agriculture and coordinator of the Biosciences Research Program at Mohammed VI Polytechnic University in Benghazi, in Morocco. Dr. Fawzi also worked as the executive director of the NRC, Canada's flagship wheat improvement program from 2012 to 2017 at the at the NRC Plant Biotechnology Institute in the field of molecular biology and genomics, at ID Biomedical in the field of DNA diagnostics, and Genome Prairie as project manager. Over the course of his career, Professor Bekkaoui has co-authored 39 scientific publications in various fields, including DNA diagnostics, molecular biology, genomics, and plant physiology. He holds 3 US patents on molecular diagnostics. So Professor Bekkaoui holds a bachelor degree in physiology from the University of Tours and a PhD in plant physiology and at the Sorbonne University, former Paris 6. So it is really a great pleasure for me to have you, Professor Fawzi, for this webinar. So the floor is yours, and after the talk, we will have a session of question and answer from the participants. Thank you.
Thank you again, Professor Aziz Yasri, for this opportunity. It is my pleasure to so if you don't mind putting your microphone on, I have a little feedback, Professor Yasri. So I have the pleasure of giving you and sharing with you some thoughts about what I refer to as disruptive technology in agriculture. So this is basically the outline of my talk. I'll go through some definition and describe what I consider as disruptive technology. I will give some example of past disruptive technology and some of the future one, and possible in biotechnology, precision agriculture. I may have missed other areas. So these are, as I said, examples. Really is just to stimulate thinking and discussions and planning for the future. I will finish with some comments, general comments at the end. So the example I'm going to give are in this area, apomixis, seed development, 2 examples, apomixis and somatic embryogenesis. I will define these later on in synthetic biologies, and I have picked 3 areas: the area of biosynthesis of high-value plant metabolite, photosynthesis, and nitrogen fixation. I will also describe the technique of genetic editing and why this could be disruptive technologies, and finish with precision agriculture. I would like to emphasize that I don't pretend being an expert of these areas. I did work during my field in the area of somatic embryogenesis. I did my PhD in plant physiology, but I'm not, again, an expert on these. I'm a research manager, and through my career, I learned with my colleagues, through the reading proposals and projects, some of these ideas that I would like to share. And these are pertinent to students that are thinking about maybe a new area of research that they may consider if they are not familiar with these. First of all, some context. As everybody knows, we face many challenges in the world. One of them is the increase in population and the need to increase food production and provide food security to the new populations. And it's FAO estimates that we need to produce, or we need to have 60% more food by 2050 in order to nourish everybody. Now, in developed countries, there is an increase in productivity estimated at 1 to 3% every year. And this increase in productivity, we can categorize that in two categories. First, there is improvement in genetics, and that represented about 50%, and the other half is about agricultural practices. I will give some example of these later. Now, these rates could double with emerging technologies, and some examples that have led to significant improvement in productivity is biotechnology, and it's estimated that in North America and in selected countries between 1996 and 2013, there was an increase in about 110 and 195 million tons of soybean and maize production. To give you some context, productions of maize in France is approximately 70 million tons. So with technologies, increase in productivity is possible. Now, the one I'm selected again, these like synthetic biology, biotechnology, other agronomic tools can address many of these challenges, faced by agriculture and food security. Productivity is not the only way to address food security. There are other ways, like reducing waste, for example, or distributing what already exists as food. So, but my focus is in relation also to increasing productivity today. So what is the definition of disruptive technology? I picked two definitions from the internet. The first one is a disruptive technology is one that displaces an established technology and shakes up the industry, or groundbreaking product that creates a completely new industry. A second definition is a disruptive technology is a new way of doing things that initially does not meet the need of existing customers. They tend to open new markets and destroy old ones. Some of these examples is the personal computer, cell phones, digital camera. On that, the disruptive technology that on the second definition, a typical example there is the iPad. I mean, no one knew about the iPad or has an iPad. And when Steve Jobs from Macintosh or from Apple's decided to build this one, it was because it was his idea. And he thought that this may be a product that will be liked by a lot of people, and in fact, that's what happens. So, but another typical example, of course, is the cell phone. And as you all know, the cell phone did not exist 20 years or 25 years ago, like now, and it has displaced a lot of technology. I myself recall using some of these tools that are shown in the pictures, like the record taper, or the paper calendar, or the calculators, for example, that are all now included within a smartphone that has replaced these technologies. Now, we come back to agriculture, and here I'm going to show some examples of disruptive technologies that have happened in the past. This figure shows the increase or the improvement of corn yield in the state, and it starts from more than 100 years ago, 1860, to approximately 2010. Or 2012 to recently. So during this time, as the figure shows, the yield has been stable for a long time, approximately 80 years, up to 1940. And from there on, several technologies have been used to improve the yield, so that the yield has been multiplied by more than sixfold, going from approximately 2 tons per hectare to more than 20 tons. And to 12 tons. The examples of technologies that have led to this are shown on the right, and that includes mechanization, irrigation, the use of fertilizer, the use of pesticide and herbicide, breeding technologies, hybrid seeds, and genetic engineering. I put these in technologies in two colors to differentiate between them. The first ones in blue are more on the agronomy side, and the second ones are more on the genetic side. And these, as mentioned earlier, combined together lead to this improvement, and maybe their contribution is approximately 50-50% each. Now, the first type of technology I'm going to talk about is apomixis. First of all, I will define apomixis, and then I'll provide the context why it is it could be disruptive technologies and what is going to replace. This is a technology that's in the early stage, but apomixis, first of all, is the asexual formation of seed from the maternal tissue of the ovule, avoiding the process of meiosis and fertilization, leading to embryo development. Its etymology in Greek is away from mixing. So basically, it's the formation of seed without fertilization, and it's shown in red, whereby a cell that belongs to the mother within the ovule that lead to a seed. And it's in a nutshell, it's cloning of the mother. Basically, it's producing a new individual seed or plant that's exactly the same as the mother, whereas through fertilization there is mixing and the new seed is different from the mother. These technologies have been developed or is being in the research stage, I should say. The proof of concept that we can make a plant that is not apomictic has been shown, and Tim Charbel and his team, and there are many teams working on this, have a patent on this technology. So why this technology is pertinent and why it could be disruptive? Hybrid seed technology, and now most of you are probably familiar with this. Hybrid seeds are seeds that are developed through breeding that have a significantly higher yield compared to conventional seeds, because the hybrid seeds have what we call hybrid vigor, and generally they grow faster, they are more resistant to disease, etc. But they go through a breeding process that's complicated and takes time. So why apomixis can be disruptive? It is because it can displace the hybrid seed industry, and if it's shown in major crops like wheat or corn, that means that the farmer doesn't have to go back and buy hybrid every year. He could use those seeds over and over for many years, and that again, this can displace and lead to a halt of using hybrid seeds. The farmer will buy once the seeds. With hybrids, the hybrid vigor is observed only on the first generation. If the farmer takes the seeds from the hybrid and plant them again, will not have the same yield and vigor that was observed with the first generation. That was the first example. Somatic embryogenesis is another example. Now, while apomixis is in the early stage of research, somatic embryogenesis is applied and can be used now. In fact, it has been used commercially in forest trees. And somatic embryogenesis is also a cloning of the maternal plant. So if we start with a plant that's already improved or has some favorable traits, one could develop somatic embryos that will keep the tissue of the mother or the parent tree or the parent plant, and the traits are kept within the generation that's planted. I believe it's also applied in oil palm, and that's done at the industrial level. So this technology could be used in more species as it's being developed with other plants. It doesn't work necessarily right away. It needs to be optimized and improved within each species through tissue culture. I'll move on to another field, which is synthetic biology. Synthetic biology is an interdisciplinary area that involves the application of engineering principles to biology. It aims at the redesign and fabrication of biological components and systems that do not already exist in the natural world, and it combines different fields from biology, system biology, bioinformatics, mathematics, chemistry, biotechnology, engineering. And I will give one example here that deals with the production of artemisinin. Artemisinin is a natural product that's used for against malaria, and sometimes there's low production of this compound, or the natural plants are not produced in sufficient quantities, or the amount produced is variable. So producing this chemical through an in vitro system like fermentation system will allow the production of higher yield and more consistent. So several researchers have developed a method of producing this natural chemical in yeast, for example. But first of all, they have identified all the metabolic pathway and the enzyme that are involved in the synthesis of artemisinin, and introduce those genes into yeast for the commercial production. So this is an example that in the area of biochemistry and genetic engineering, this could be applied to many other chemicals, and examples of course, where this could be applied. There are possibilities of producing a rose smell variant in yeast. Cannabis THC, tetrahydrocannabinol, can also produced in a synthetic way. And again, there are many other examples. I mentioned here artificial chromosomes, which are again DNA in a form of circles that could be manipulated and introduced into cell culture to produce compound from whole biosynthetic pathways that could be introduced again into plant. In the area of synthetic biology, there has been a demonstration of the whole biosynthesis or development or preparation of a bacteria, mycoplasma, which has been produced. And these cells also can be used to produce specific compounds in the future. Another example of disrupting technologies that could be used in the future is what's referred to as plant Sentinel Biosensors. Basically, an entire plant is modified to detect and signal the presence of specific component in its immediate environment. So the plant can be programmed to respond to a wide variety of environmental pollutant, nutrient, abiotic stress, and other environment. I mean, a typical example here is in a field whereby maybe the composition of the soil is not homogeneous, and we put some plants that are modified to alert the farmers of the need of some compounds. The example that has shown is the possibility of detecting basically deficiencies in phosphate, and scientists have modified Arabidopsis where it is starved from phosphorus, and when there's a starvation, the plant turned into blue. Here they use the marker, which is beta-glucuronidase that makes specifically a blue compound. This is an example in principle, but could be applied to many others. And the advantage again here is to help the farmer identify stresses or deficiencies before they become to have an impact on the yield. An example of disruptive technology is the modification or the improvement of photosynthesis. First of all, I'll briefly describe photosynthesis. I'm sure most of the people in plant biology are familiar with photosynthesis, which is basically photosynthesis drives agriculture, and it's the sole defining and unifying feature of green lineage organisms. Plants are required to have chloroplasts, rubisco, which is a protein or enzyme that involve in photosynthesis and have chlorophyll. to make sugar and oxygen with starting with CO2 and water, and of course the involvement of sunlight. So, but photosynthesis is inherently not very efficient. It has a maximal theoretical efficiency of 11%, but most of the plant, the efficiency is 1 to 3%. And if we can improve this, let's say going from 2% to 4%, it's almost like doubling the productivity of a plant. This is a theoretical, of course, assumption, and it will for sure require a lot of research to get to this point. There has been some proof of concept and some work with cyanobacterial enzymes, and shown that it is possible to improve photosynthesis. There's another example where we could improve productivity through changing photosynthesis, and that's in the case of what's referred to as C3 versus C4. C4 plants, like maize, for example, have a higher efficiency of photosynthesis because they have a specific enzymatic pathway, have a structural structure that allows them to produce a more to be more efficient. And knowing those pathways and transforming those pathways into other non-C4 like wheat or other plants where the yield is comparatively low to maize can potentially improve also the efficiency of photosynthesis and the yield of crops. A very important trait is nitrogen fixation, and here again, producing plant with nitrogen fixation could be disruptive because it could displace the area of fertilizer. First of all, nitrogen is a bioavailable form of crops, is very expensive to produce, and it basically it uses right now 1% of the total world energy expenditure. Most of the plant can't fix nitrogen except legumes, which have rhizobia on them. So legumes are able to fix nitrogen, but not the other crops, which are the majority. Some just definition or reminder of nitrogen fixation. Basically, which occurs when atmospheric nitrogen is converted to ammonia by an enzyme called nitrogenase. The formula is shown in here with the nitrogen in the gas form with ATP. It leads to ammonia and ADP and phosphate. So as I said, the legumes family is one of the unique plants that are able to fix nitrogen because of rhizobia. And when the plant die, the remaining parts that stays in the soil, they provide nitrogen that can fertilize the plants that are going to come after. Generally, legumes are grown in rotations. So first of all, a farmer can plant legumes with very little or no nitrogen, and then after the legumes, the farmers can plant, let's say, cereal. After legumes, generally the cereals don't need as much nitrogen because of the matter that's left by the legumes. Now here there are several companies that are working in this area, including Ginkgo Bioworks and Bayer and Joint Bio, and they are basically targeting to reduce the global fertilizer by 1/3. I switch gear here to completely different technologies, and here it's genome editing. Probably most of you have heard of this technology. I believe it could be also a disruptive technology that could displace the way breeding is done conventionally. It's still in its infancy and require a lot more research to get to that. The picture here show a baby that had a serious disease. This was in 2015. This is an article from a BBC website. And this baby basically, according to the doctors, could not be cured because could not handle a specific drug. Had a form of cancer and was basically dying. And it was the early stages of gene editing, and the doctors proposed to the family trying this method whereby they will have to transform a gene and change the biosynthetic pathway or the biosynthesis of one compound in the blood of this baby. And fortunately, the technique worked through gene editing, and basically the baby was able to accept the drugs and survive. And the last time I checked, the baby, I believe, is doing fine thanks to gene editing. How gene editing or genome editing works, an enzyme is required. This enzyme has a long name. It's called CRISPR-Cas, which stands for cluster regularly interspaced palindromic repeats. And the enzyme require a guide RNA, which is shown in purple in this picture. The enzyme is shown in green. And through the guide RNA in the enzyme, it's possible to change the original DNA, which is shown in red, and make minute change, which could be deletions or insertions, a modification of the original DNA that could lead to a new trait, and which was done in the baby as shown. Basically, a nucleotide has been changed so that the baby can be more tolerant to the drug. This method is precise, efficient. It's not exactly a transgenic method if we take the classical definition of GMOs, because we are not introducing foreign genes. And I would say that it's comparable to mutagenesis, which is used in conventional breeding. And it has tremendous potential. In the last 5 years, several examples have been shown. But with the improvement of the efficiency of the method, again, it could be quite a quite transformative methodology in the future. Biostimulants is another area that could displace several industries or type of product that are used now, such as chemicals, hormones, etc. First of all, the definition of biostimulant is means a material which contains substances and or microorganisms whose function, when applied to plants or the rhizosphere, is to stimulate natural processes to benefit nutrient uptake, nutrient efficiency, tolerance to abiotic stress, and or crop quality independently of its nutrient content. So it can cover several fields of agriculture. A typical example, let's say a microorganism that could be considered as biostimulant is mycorrhiza, for example, that could help the plant absorb and more efficiently phosphate or solubilize phosphates, and thereby reducing the use of additional fertilizer. There are some applications now, but with the knowledge of more microorganisms that are in the soil, it's likely going to be an increase in technology that could benefit to the farmers. Up to recently, we knew very little about the microorganism in the soil. With new methodologies like genome sequencing, soil testing, it's likely going again to lead to more biostimulants and microbes that could be used by the farmers in the future. So now we are going to talk about precision agriculture. Now the biostimulant and precision agriculture are more in the area of agronomy, whereas the technologies like gene editing or genetic modification are more in the area of crop improvement or genome or genetic improvement. Precision agriculture is an area that could be applied to improve basically practices. So the definition is precision agriculture will aim to make decisions for farm management with the goal of optimizing return or input while preserving resources. And some of the technologies include satellite positioning system, remote sensing devices, diagnostic tools, and delivery devices. So the farmers would be able to use this technology to detect evidence, for example, of drought or pests, infection. Information about these conditions would trigger an automatic adjustment of irrigation level or pesticide application. A typical example in here could be the use of a drone, which the farmer could use to monitor for example, the presence of a pest, and following that detection could trigger application of pesticide or biostimulants or a product, natural or synthetic, that could be used to stop the pest before it goes in the whole field, and thereby reducing the amount of input, reducing the costs, and preserving the environment because the less chemicals have been used. An example I gave the example of the drone. Another example that could be foreseen in the future is a weeder. As you may know, herbicides are used widely to remove weeds that competes with the plant and reduce significantly the productivity and the yield. A Swiss company, Eco Robotics, has developed this tool that has a detection level of 95%, more than 95%. Basically, it will discriminate between the crop of choice, the one we want to produce, versus the weed. So it has a camera that can, again, take pictures and detect. So in the typical scenario, without the precision agriculture, one would use herbicide widely all over the field. Here, because we are discriminating between the weed and the plant, we will use herbicide, but we will target only the weed. And in total, well, at the end, 20% less herbicide can be used. An estimation cost 30% less. This will, of course, so no herbicide left on the crops of choice that we are going to eat. It will conserve also the organic life of the soil, and it uses solar energy to power the robot. So those are, again, some examples. I mean, I'm sure there are others. It was, again, just to give an idea here how one could foresee some of the future applications. Some of these technology are in the very early stage, are in stage of research. Some are very close to application or even started to be applied, and these will again displace some of the current technology. Now, how this could be in the future? I mean, one can foresee a farm in the future that could be completely different to some of the farms now. I could foresee, for example, a farmer starting their day instead of going in the field and walking and watching what's going on at the beginning of the day, maybe they'll start their computer and checking the weather, checking if it's going to be raining or not, checking if there's any threat from some pests, from information from the neighbors. And if depending on that information, they may decide to send drones that's going to be in their garage, do a tour of the farm, taking pictures, sending the pictures to the computer, analyzing the data, and depending on the results, maybe sending another drone with a biostimulant or bioherbicide or bioinsecticide that could treat a very local region instead of treating the whole field. They may send a robot to do weeding, and they will also share their observations with neighboring farmers through their computers. All of these could lead to reduced use of chemicals or intrant and reduce the cost. Farmers still needs to go to the field and walk in their fields to see, and hope they'll do that to see by themselves and confirm their observations. This is an example of what could be the farm of the future. Now, will this be accepted by the public? Will some of these technology be accepted? I gave, in my view, there has to be certain criteria, and the first one is the technology has to be durable. I mean, it has to show that it's not going to affect the environment, the health of consumers, for sure. The costs have to be reasonable. Some of these tools are going to be expensive in the beginning, and depending on the size of the farmer, some may be able to apply them, some may not, and has to also lead to more revenue for the farmer and to be more beneficial to the farmer, of course. That concludes my talk. I took a lot of information from publications and the internet. Some of the references are shown in this slide, and I would like to thank you and welcome questions. Thank you for your attention. Professor Aziz Yasri, are there any questions?
Hello, Fawzi. Hello again. Thank you for this presentation, Professor Fawzi. I'll just turn on my camera. So for this is a really nice, interesting presentation, Fawzi. It was very informative. Just for the people who joined just a little bit late, this webinar, I'd like to say that you can still watch this presentation made by Professor FAOUZI BEKKAOUI from INRA through the ESAFE channel in YouTube. So all the presentation is recorded, and you can watch it again and again anytime you want. So if you have missed a part of it. So now it's time for the questions and answer session. So I will, since there is a lot of questions, I will start with putting them one by one. So I will start with one question from Professor Michel Ghanem. Professor Michel Ghanem is from the University Mohammed VI Polytechnic, a colleague of mine. Michel is asking, he's asking you, Professor BEKKAOUI, what is your opinion if increasing photosynthesis is a likely solution for the world food problems?
Thank you for that question. Again, I mean, there are many institutes and research groups, programs working on this area to improve the rate of photosynthesis and contribute to world food problem. I would say that it's one possible solution. There could be many other solutions to increase productivity. To say that only photosynthesis can do that is likely not correct. But we have to survey and test all options. I mean, apomixis could be one of them. Precision agriculture could be one of them. So all of these are new tools at the research stage that we have to test and try, and effectively increasing the rate. In theory, I mean, if we compare wheat to corn, although I mean there are two different groups of plants. One has a higher rate of photosynthesis, and corn produce a lot more seed per acre or per hectare than wheat, because their rate of photosynthesis is higher. So in principle, why not? But that will not be demonstrated until we do the work. And again, photosynthesis could be one of the solutions.
Thanks, Fawzi. So before the question of Michel, he made a comment. I'm sorry, I didn't show it to you, saying that photosynthesis rate is not limited to crop and yield. Without enhancing nitrogen uptake, storage, and transfer, there are now physiological paths for increased photosynthesis to enhance crop yields. That was his comment before the question about. But I think you have answered his question. Okay, so we have a question from Nuyifoni Laurencia. She's thanking you about the presentation, and she said, what are the indicators of agricultural system disruption technologies?
Well, I would say that the indicator is when the original technology is not used anymore or displaced. The typical example again is the regular camera replaced by the digital camera. So for a long time there was only one type of camera. When the digital camera came in, it displaced, and now nobody used the old type of camera except maybe some professionals, and even those are using digitals now. So in the area of agriculture, again, there are many examples. One of them could be the biostimulants. We use now a lot of chemical, artificial chemicals that are used. We know that nature, or there are some potential stimulants that could be used. Once we identify those and optimize them, they could replace the old chemicals and be again an indicator of a disruptive technology. I see another comment. Apomixis could be the key to preserving the seed yield gain of hybrid from one generation to the next. What is the current status and innovation in this regard? I'm aware of one team, and the best thing is to refer to go directly to this team and ask them. I'm not myself an expert or doing some of this research. The team I'm aware of is the team of Tim Charbel, who's working in Saskatoon at the Global Food Security Institute. They have done a lot of work in this area, and to my knowledge, they have some proof of principle. So I would invite the person who asked me to check their website, contact Tim Charbel directly, and to find out what's the status. And I'm sure there are other teams. The proof of concept is encouraging, but we are quite far from the applications of this technology. But again, this is also a typical example of disruptive, because if it works, well, there won't be any hybrid seeds in which is a approximately $40 billion industry, which is quite significant.
Okay, Dr. Professor Fawzi. So we have a question from Adnan Beniyash. The use of disruptive technologies is based on databases. In Morocco, we have the problem of data availability. Does INRA have any strategy for sharing agronomic data?
Interesting question. We should, and I mean, INRA publish its work through publications, through books, through fact sheets, etc. This gives me an opportunity to share with the participant that one of the journals of INRA, which we call AfriMed now, which was called Al-Awamiya. AfriMed is African and Mediterranean Journal of Agriculture. and this journal will share INRA's data. The issue we published just came out is on pasture land, and the next issue will be on citrus research. So this is research of INRA, and these are examples. And I'm optimistic there will be more other data shared. That is said, do we share all the data? Probably not. Probably there is some data that stays within the scientist, or maybe because it's not relevant or it's that couldn't be published. We need to be more organized and more developed and gather that data and share it in a more efficient way. And this could be done through different ways. I mean, it could the most simpler is through partnership and collaboration. So people are invited to talk directly to scientists from INRA and find out what data they have and how they can share that data. And the other through a website database. And I'm looking forward to sharing more of this data in the future.
Thank you, Fawzi. So we have a question from Goni Shirinda. He's thanking you for the presentation. He said it's very informative. He's asking you, do you have examples for potential disruptive technologies that did not become mainstream? What went wrong? What lessons can we learn from this, if you are aware of?
Well, I mean, research is by definition is a very high risky business. I mean, you just have to look at the patents, and a patent is by definition is something that's innovative, that's new, that's not obvious, and in principle maybe every patent could be disruptive because it's made to make a product, a commercial product. And maybe in patents, only 1% will end up being commercial. So I would say that there are a lot. I'm aware of biotech companies because I was in this field that started a business and never made it commercial. So yes, there are a lot, but I'm not going to mention them. But I mean, maybe to give an idea for this person who asked, just look at the patent and go to a database and look at how many patents and how many of those that are really commercial. So I would say relatively a small proportion of the research make it to commercial. But that's the nature of research. I mean, you have to do it. When you have an idea, a hypothesis, you have to do the research to find out whether it's going to lead to a product or not. Now, there are sometimes very good ideas, very good product, but does not make it commercially, not because it's not an efficient product or it's not a good product. It could be due to management, it could be due to lack of finance, it could be done through other social or political reasons. So some technology don't make it, again, because of other reasons rather than the technology itself.
Thank you, Fawzi. A question from Abdulaziz Ghazali. He's thanking you for the presentation. And he said, at this stage, can we rely blindly on precision agriculture in determining nitrogen to apply for crops?
I don't know why the word rely blindly. That kind of defeats the purpose of precision agriculture. It's not a blind. If it was blind, it should not be used. Precision agriculture is based on analysis, and based on that analysis, apply a certain amount of nitrogen. I'm not an expert in this technology, but I'm aware that it started to be used in quite a bit in North America and maybe other countries. So it could be that we need to validate some of these methods. It's at an early stage, but I think it's a very promising technology and based on facts. So if you have a soil that doesn't have enough nitrogen, that means you need to add more, and that is determined based on the initial analysis. So that that would be my answer.
Thank you, Fawzi. So we have a question from Samuel Ajele from Futa in Nigeria. He said, disregard to nature in application, whatever this technology, smartness, efficiency, would ultimately cause disruption of the delicate balance of natural ecosystem. What do you think about these comments?
Well, I would not, again, there's a word that I don't agree with, is all technology will ultimately cause disruption. I mean, gave one example which will lead to resolving a problem of durability. In fact, in my conclusion, I said that for these technology to work, they have to show a minimum or three things. One is they have to be durable. So in other words, they should not disrupt the environment. And when I gave the example of precision agriculture, I'll give two examples. The robot that's going to replace the wide use of herbicide, where we are going to reduce 20% less herbicide. For me, this is going to improve the situation rather than make it worse. The other example is the biostimulant, where we may go from a chemical stimulant to a natural stimulant. These are new technologies that are in the early stage that can disrupt but solve a problem rather than make it worse. Now, the other option is if you don't want to use any of these technologies, then and we do need to improve productivity. I'll pose a question: what other alternative do you propose? If you have a technology that can improve productivity by 60% in 20 or 30 years, then you should start a research or propose it in a proposal and give that as an alternative. I'm aware of several technologies in the area of organic agriculture, agroecologies. Those are very good ideas and technologies, and those are more durable. And I think they need to be validated, demonstrated. And if they show that they are durable, why not? We'd use these technologies, and if they can satisfy food security, they should be used. But I don't think we should limit our application only to those. My understanding is the estimation of food production now is that 50% of the food we produce now is due to fertilizer. So if tomorrow we stop using fertilizer, we'll have 50% less food. Then I'm asking the person who asked the question, what do you propose? Thank you.
Okay, thank you, Fawzi. Here's a question about the economical side or policy side. From Malika, Madam Malika Abu Hussein. She's thanking you for the topic. And she said, what about the policy and institutional regime and resource conditions? Do you think that we have all of this to enable disruptive technology?
Well, I mean, it depends on, I think it probably will be case by case. And we definitely need the institutional resources. We need the legal aspect to follow on these technologies, and probably be case by case. I mean, a typical case is the drones. There are some institutions in some countries that don't allow the drones because it can interfere with airplanes, with privacy, whatever. So we need to go to the legal stage before we apply this technology, and it would be case by case. Sensors to measure the water content in the field, they're already applied. So probably you don't need regulations for that. Genetic engineering, you need regulations, and those are complicated processes and need a lot of time. And but once the technologies are validated and demonstrated that they are safe, durable, the widespread use is probably okay.
Okay, so we have a question, I think, from someone, Mabuva. I don't know the name, but probably it's Professor Abul Berbah, because he's using probably using Mabuba. He's thinking about the presentation. He said precision agriculture, is it reality in Morocco, or we still so far from that? Conservative agriculture, organic agriculture, precision agriculture, which one is more suitable for Morocco?
First of all, is it a reality or not? I mean, I visited the farm that use sensors to monitor water before applying water. So there are cases, it's not widespread for sure. INRA has done experiments with the using drones to deliver herbicide in a field of rice. And that allowed to use a lot less water, a lot less herbicide because it's more controlled. Those are experimental. But I think we will use these technologies more and more. Now, which one? Can you go back to the other one, the other question? Anyway, which one is more better for Morocco? I don't know. I mean, if we can exclude one over the others, I think it has to be case by case. And organic agriculture is priority area for the new research strategy and for the new Moroccan plan, the Generation Green. And it's an area that we are going to develop. INRA is going to have a significant activity in this area because there is an opportunity. There is an opportunity for the farmer, for the consumers. There is a demand, and we will do and work on these. But there is also conventional farmers that still use pesticide, herbicide, but we would need to reduce the amount of these chemicals that may have a negative impact on the health or in the environment. And here, precision agriculture could be also used. So I don't think we can generalize and say only this is good for Morocco. I think it will be case by case, and depending on the consumer, depends on the environment, and we need to work on all of these for sure. And with INRA, we are going to work on all of those technologies.
We have a question from Professor Lamari Abdelali. So, you know this person?
Yes, and that's a very good question. Actually, I was thinking to say a few words on this, but this is an opportunity to address it. Yeah.
Yes, the question is about
go ahead.
Adoption of this technology by small farmers.
Yes, of course. Now, again, it depends on the technology. I mean, apomixis is not going to be happening in a year or two, or maybe even 5 years. So apomixis, we are talking at least 5 years, in my estimation. And will the small farmer, I'll take two examples. I will take one, it's long term. Apomixis, small farmers will love this technology because he or she will need to buy the seed only once, in theory. Whereas now, if the small farmer is using hybrid tomatoes, they will grow the tomato the first year. They need to go back and buy the seeds the next year. And so apomixis in the long term, it's going to benefit the small farmer. Second example is precision agriculture and use of modern tools. And obviously, I mean, the average size of a farm in Morocco is maybe 2 hectares, and revenue is relatively low in small farms in Morocco. And buying a drone or using sensors, it's probably not going to be possible in the beginning. What may be possible, but first of all, we need to validate these technologies in a small scale, and INRA and its partners need to do this type of experiment and see if we demonstrate that the use of drone is beneficial. We need to do estimation and see how this is going to, whether it's going to be cost effective. So again, a small farmer will likely not be able to afford a drone. But what about if you put now 10 farmers in a cooperative? And now here again, we need, and you as an economist, Dr. Lamari, could help in doing this calculation and calculate for 10 farmers or 20, whether sharing a drone will help them reduce their input and improve their costs. And those are the type of work that we need to do and validate. I believe in the end, it's probably going to be beneficial. Similarly, I mean, precision agriculture is also data analysis of weather or other large databases. And here also, the farmer alone and the small farmer in Morocco probably will not be using this. I mean, they are using smartphones. They start using smartphones, and they could access that data if they are formed and trained. But the analysis of data probably has to do with another partner, whether a university or research institutions or some of the institutions that are helping farmers to transfer technologies. So the adoption probably going to be small in the beginning, but increasing with the reduction of costs of this equipment. That's the law of scales. The computer, when it came first, was very expensive. Only a few people can afford it. After several years, more productions of computers, the cost went down, and it would be similar in agriculture, in my view.
Thank you, Fawzi. I know that you haven't touched on the GMO, and so there is a lot of question about this. One of them is this one, is from Dr. Rida Ben Mri. He said, as genetic modification of plant is prohibited in many countries, what do you think of making genetic modifications of microorganisms that interact with these plants, fungi, and bacteria to make them more effective?
Okay, it's a very delicate and sensitive issue, of course, and there's many different views on this subject. If we take two examples of North America and Europe, for instance, where in the first genetic modification is allowed and some products are being commercialized, and have shown in the last 20 years that they have, compared to conventional agriculture, no negative effect on the environment and health. And these countries are going to continue using these technologies. So these applications that the person has asked the question probably are going to be used in these countries. In Europe, the question is different. The political system has not allowed the use or widespread use of genetic engineering. Where I see a future, and that's why I gave genetic editing as an example. Probably the classical way of genetic engineering is not going to be used or not going to be encouraged. What I mean by that is the transgenic, whereby we are taking a whole gene from one species and putting it in another species. Even though this technology has not shown a negative effect on health or the environment compared to other technologies, it's still not accepted in several countries. On the other hand, genetic editing, where you are staying within the same organism and just making one or two single changes, in my view, that technology is very similar to what's used now in Europe. Mutagenesis that's used in Europe is effectively a genetic editing method, except the way to use it is different, but the end result is the same. In fact, genetic editing is better because it's more precise. I notice, and I think some people do not differentiate. Well, look at more at the process than the product. In my view, we should focus on the product. Right now in Europe, there are products that are generated through mutagenesis that are similar to what would be generated by genetic editing. However, the rules and the regulations don't allow genetic editing, whereas they are allowed mutagenesis. And it's a fundamental contradictions in my view, because we are looking at the process rather than looking at the product. So, and in conclusion, the legal aspect needs to evolve, and these technologies will probably be more applied and be beneficial in the future. I can hear you, Professor Aziz. Yes.
The other side of the question was, instead of editing the gene of the plants, could Reda Ben Mrid was suggesting, could we act on the microorganism by editing microorganism that can help the plants and improve the plants, for example, yield and the growth?
Why not? I mean, it depends on the case. Why not?
Okay.
If we know the mechanism, and yeah, but I mean, it has to be demonstrated that it will not have a negative effect on the environment and or an obvious effect. Yeah, I think it's in my view, it's easier, better to do it on the plant because you can control the plant better than microorganisms. But why not?
Okay, so we have a question from Muhammad Amari. He said, in your opinion, which of these disruptive technologies that can have a high added value / cost ratio, and that can help Moroccan agriculture facing most of its recent challenges? So he was talking about the precision agriculture, the drones, the gene editing.
Well,
which one of these?
Yeah, I mean, it's very clear that genetic editing, photosynthesis improvement, or apomixis are technologies that are in the research stage. I mean, I picked those because we have a lot of students, well as and PhDs or masters that could be interested in exploring this technology and doing some of the research. So they are not going to be applied tomorrow or even in the next 2-3 years, maybe even 5 years. So these are research. Genetic editing, we can't do it in Morocco. We don't have the legal aspects to work on commercialized genetic editing. So definitely a no at this time. On the other hand, we could do research on those. Now, in terms of application, the one that I foresee being used in a relatively short term are precision agriculture. The sensor, the use of sensor. I mean, water is a very important factor. I mean, everybody in Morocco knows that the year it rains sufficiently, we produce a significant portion of our cereals. The year it doesn't rain, we have to import more cereals, and the economy is affected. So water is very important. We need also to preserve this resource. In some areas of Morocco, overuse of water has led to the dry, to some wells being dry and affecting the agriculture. So the use of precision agriculture here is key. By how? By using sensors that can monitor the amount of water that the plant needs and giving only what the plant needs. And very likely that these technologies will lead to conservation or better use of water. There are other ways of doing that, but the choice of course of the crop is very important. But that will be one example. The use of drones, and here again it'll be case by case, where we are giving pesticides or herbicide or biostimulant rather than using a conventional method, could also be used in the short term. So in summary, the precision agriculture tools will probably be the one used in the near future. The other ones, we are talking several years. They are more of interest to research at this time.
Thank you, Fawzi. I think we were interrupted. So we have a question. I'm sorry. Thank you, Professor Fawzi. I was my microphone was off, and the echoes. This is why I turned off my microphone. So Mohammed Boulif, Professor Mohammed Boulif, for this presentation and the nice talk. He would like to know your opinion of Generation Green program in terms of creating jobs and wealth in rural areas through IT?
Thank you for these questions, Professor Boulif. It is very pertinent question, and indeed, Generation Green is going to give a big emphasis and priority to the use of IT and digital agriculture in the future, and probably will create jobs and wealth. I mean, again, there are many examples that could be used. I mean, the farmers are probably going to be using more and more their smartphones or computers, for those who have computers, to maybe sell their products, check for prices, borrow money through banks and banking, online banking. And I'm not familiar with all the programs, but I'm aware that the Generation Green is going to give an emphasis to this area, and I'll be happy to share in the future some of those ideas as I know more about them. But the Ministry of Agriculture is going to talk and share some of these programs in the future with the farmers. And again, definitely it will play an important role, IT, in farming in Morocco.
A question from Professor Jamal Ourak. He's thanking you for the presentation, and he's asking, most of the technologies you have presented need more research. In which field do you think that INRA and other Moroccan institutions can be leading in order to focus on them more?
It's a good question. We are in the process of developing our program for the next 4 years, and I would say that the precision agriculture is probably going to be one of the areas that's going to be more of an emphasis. Some of the biotechnology areas may be planned. We are not planning to work on photosynthesis at this time, or apomixis, so I can say right now. Somatic embryogenesis, we have some activities on this, in particular on argan. Argan tree is very difficult to multiply vegetatively, and somatic embryogenesis could be one of the methods that could be used. So some of the colleagues are working on this area. Synthetic biology, we're not working on this area. I shared some of these ideas because of my previous jobs, and I thought I'm sharing with them not because we are doing them at INRA, it's because our ideas that I would like to share with students for discussions, for thinking. So in summary, the things we are working on is we will be working on essentially our precision agriculture, somatic embryogenesis, and classical improvement in the use of markers. I didn't talk about that, but those will be speed breeding. I didn't talk about some of the methodologies we're probably working on.
Yes, we have a question from Professor Khalid Ofdou. He's thanking you, Fawzi, for the presentation. He was asking if he was wondering if the focus on selecting more native tolerant plant varieties and genotype is better than using genotypical changes that can disturb the environment.
Interesting question. I'm going to describe, I have to go back and describe the classical breeding, the way, I'm not breeder, but the way I understand it, and I'll describe the genotypical changes. The classical breeding, if we take an example of, let's say, wheat that's become sensitive to a fungus, the way we would improve that wheat, classical way to improve that wheat, is by doing crossing using a germplasm from a gen bank or a native race that has the resistance, but maybe does not have the quality or the yield. So the breeders will do crossing to introgress that trait into the new variety. In conclusion, that breeder has changed its germplasm or its plant to introduce a new variability, new variation, new allele. So there is a change even through classical breed. And if we are looking for yield, if we're looking for staggering traits, if we're looking for specific quality, we have to go through this process. We can't go into a native plant and expect to have all those traits. And native plants may have some very good properties. Let's say a plant and medicinal plant. If there is a plant and medicinal plant that exists in nature and has some product, and we're happy with the amount, with its agronomic values, but we can produce it. But now if we want wheat to be resistant to a fungus, or to be resistant, to give more yellow color, or to have a higher yield or more drought tolerance, we can't just take a wild plant and expect to have those same characteristics. So we have to go through conventional and classical breeding. And if we go through that, we will be generating some changes in the line we are making. What genetic editing is going to do is essentially it's going to do the same thing, except instead of going through those crossing, we are going to go back to the native land, that the native lane that has that resistance, identify the gene, identify the variability that's giving us that trait, and doing it in our plant of choice. So we are going to skip, in theory, we are going to skip a lot of work to arrive to the same product. So again, I go back to this, we have to start thinking about product and process. If the product is, because the product is what matters, is the product what we're going to eat, is the product that's going to be in the environment. Forget about the process if the result is the same. If there are some philosophical reasons why that process should not be done, that's another thing. But on the scientific, on the basis of science, on facts, for me, it's the product that matters, not the process. So I don't agree with so.
Thank you, Fawzi. As we are approaching now the end of this webinar, we will just probably trade like 3-4 questions, Professor Fawzi, if you allow me, and then we'll give you the last word at the conclusion for closing the webinar. So one of this question, this last question, is from Madame Taimi Nawal. Thanking you for the great presentation. She said, with enhancing photosynthesis efficiency, will we really get more green yield, for example, or will the plant just keep producing more leaves and stems? Very technical questions.
I'm again not an expert in this area, but the idea is to produce more grains, of course. In order to have better photosynthesis, probably there will be more leaves and stems, but the energy has to be directed into the grain too to get more yield. So I expect the researchers will choose the plants that will have both, that will increase photosynthesis, but also will increase seed and yield. Otherwise, we wouldn't do that, except if we are producing biomass for other applications. But if we want to produce grain, of course, the photosynthesis has to lead to more grain. So, and it's possible, I think. So I can't go into more details. I'm not an expert in this area, but it's probably going to be possible.
Thank you, Fawzi. So a question from Sefemi Femi is Nigerian. I know this student is from the Fertilizer Science and Technology master degree that you have. He's continuing, and he's asking, how do we canvas for adoption of disruptive technologies, considering the cost and scale of this technology in Sub-Saharan Africa? Economical question.
Mm-hmm. It's a good question, and probably the first thing that in my view, we need to do is to validate some of these technologies, demonstrate on a small scale whether this is going to be beneficial, whether the cost. I'm sure they will be beneficial. The question is whether the cost are sufficiently interesting to apply them at the small scale, and probably take more research and more improvement. Probably the tools would have to be not expensive, or they have to be low enough for countries in Sub-Saharan Africa to be used. Now, one of the maybe the first applications in Sub-Saharan Africa is in terms of soil analysis and benefiting from the use of fertilizer to increase the yield. And I think there are some countries that have shown I think in Ethiopia, where they've done in some areas soil analysis and modified the type of fertilizer they use, has led to improving significantly the yield of crops. So I think other African countries could benefit from this. So they are not, maybe the small farmers would probably not be have the tools and the money or to do soil analysis, and that's where maybe government, other industries, associations can help do some soil analysis, whether it's through satellite imaging or through drones or through some scanners that will pass through the soil and do some quick analysis, and then give a formula to the farmers to use that's more adapted to their soil. So these are some examples I could foresee being applied the sooner, that could help the farmers in Sub-Saharan Africa. The other technology, as mentioned, they are, again, research areas, they are long-term applications.
Thank you, Fawzi. We have a concern and a question from Mr. Gad Matayu. He's thanking you for your presentation. Suppose I have greenhouse as other type of disruptor technology that can easily be adopted by small-scale farmers. Why haven't you included the here? He's referring to this very small disruptive technologies that are today already adopted by some of the farmers.
Well, I mean, greenhouses have been used for a long time. Maybe it could have been used as an example as a past disruptive technology. It's not something very new. So that's how I would see it. Now, what could be disruptive is there's some background. What could be disruptive is maybe hydroponics that are used within the greenhouse, or some other technologies. But the greenhouse alone, it's not a future disruptive technology, because it's not new. Your microphone is.
It's a question after you said about to focus more on the product rather than the process. As is from Mr. Anthony Oyebe. He said you highlighted the need to develop the product rather than the processes. Don't you think we need a model to guide smallholder farmers for wider adoption?
Or maybe I didn't explain better, or I think both are important. We need to develop the processes. When we talk about apomixis is a process. When we talk about genetic editing, is a process. What I meant by product versus process is we should not judge a product based on the process. We should look at the product, its impact on the environment, its impact on the health, not the way it has been developed. but both need research, both need development. So that's I think where I mentioned that how to guide. We need a model to guide smallholder farm. Honestly, I don't understand this statement in relation to product versus process. So.
Okay, so Fawzi, I'm now summarizing a bunch of questions. It's about this gene editing technologies in plants. So they are asking about to what extent the results of the gene editing, I mean the products, the plant, can be somehow patented through IP, to intellectual property management.
To what extent this could be patented?
Yes, the result of this gene editing, the product itself.
Mm-hmm.
Let's say,
well, the product.
Go ahead, please.
So the process, like the gene editing method on itself, so that's a process that was described is already a patent, and there's already a patent by 2 institutions in the state. So that's one thing. So if anybody wants to use commercially gene editing, they would need a license from these original people who patented the process. If it's for research, I think it's different, or they need a research license. Now, if one has to apply that on, let's say, on a crop specific to their countries, it probably be case by case. I mean, the things that guide the process of patenting are novelty, commercial potential, and non-obvious. So I'll give an example. I'm aware, I know, I worked on it, is the herbicide tolerance in canola and the modification of ALS acetyl-lactate gene. If somebody wants to apply this in their crop, which has already been done in others, it's probably been very difficult to patent, even by gene editing, because nothing is, because it's not obvious, because it's obvious, and it's already known. If they find some new idea that has not been applied, then probably it'll be possible to patent that.
Thank you, Fawzi. We have a question from Professor Tanji Abbas. Thanking you for the presentation. He said, do you think that Morocco has enough human and financial resources to do this type of research on priority crops like cereals or legumes?
Interesting question. Now, and I mean, I speak of INRA. We can't do everything I described. As I said, I presented this as a way to initiate and discuss some of the ideas with students, and because of the job I had before, and I learned about these technologies. For Morocco, for specific technologies, like I said, like precision agriculture, INRA will be doing some of this work on precision agriculture, on tissue culture, somatic embryo genesis, for example. We do have some expert in this area. We will work with partners on precision agriculture. We will work with partners. We have a couple of small companies we sign an agreement with that are expert in drones, are expert in the use of data, and we will do that. So, and we do have our own resources to be able to do that. Do we need more resources and more human resources to do more? Definitely, yes. But I would say at this time we can do at least some of the parts that I described on precision agriculture. Doing research on apomixis or disrupting photosynthesis, I think those could be ideas for PhDs. Why not? They need to find the right professor who wants and interested to explore those ideas, and they are risky, very high risk. If I was a PhD student starting my doctorate, why not? It could be an excellent subject of a thesis. And now, do they have the financial resources? It depends. I mean, they have to maybe work with collaborators in other countries. They have to have a fellowship or a scholarship, and they have to have a minimum of tools and ways. I can't speak about all Moroccan universities, and probably some don't have the resources. But there are also grants. People can apply for grants and seek funding through this way. But one thing I like to say is people should not be discouraged because these technologies are sophisticated. Therefore, we should not do them. I think through partnership, through applying for external funding, maybe it's possible to explore these areas.
Okay, thank you, Fawzi. That was the last question for you. So as usual, we have now to answer some of the questions of the students. This is for me. Some of I'm sorry, it's not this one. It was a student who was asking about a certificate of participation. Of course, we could give you a certificate, no problem. So if you can send us the request by email, we can send you a certificate of participation to this webinar, no problem. So there is other questions, Fawzi, about the research in this area, and this is from PhD students or master degree students. So they are wondering what are today the key area in this research domain that they should focus on, to bring new solutions and probably to make PhD projects for their thesis.
Well, I mean, all of them. It depends on the supervisor. I mean, they need to have a supervisor that's either an expert or would like to explore these new areas. So that's the first thing. You can't just start one of these subjects anywhere without an expert or a supervisor or professor that's interested to explore these technologies. That will be the starting point. After, I think as a PhD, I think students should just come up with a good hypothesis, and any of these could be the subject of a PhD. You don't have, in a PhD, I think your main objective is to understand and generate new knowledge that's new. It does not have to be commercially applicable right away. The key is to come up with new information that will allow you to publish. Could be patent if you want. If it's you may end up with something applicable, but that's not the main objective. So again, you have to find a supervisor that can guide you and do. I mean, I can't tell you to go in this area or others without having the supervisors. I mean, it's basically them that will have to help you, guide you there. Precision agriculture, there's a lot of progress there, and digital agriculture, and those probably will be more applicable in the near term if you are looking for something more applicable. And there's a lot of ideas and potential in that area.
Okay, thank you, Fawzi. Thank you again. And the name of agriculture, ESAFE, the University Mohammed VI Polytechnic, and the name of our strategic partners, OCP-Africa and RUFORUM. We would like to thank you for the time you spent with us and for this very interesting talk, and also for having answered the majority of the questions that you have been asked. Thank you again. So you are more than welcome to come back with any new subject you would like to talk about. This is a continuous series. We'll have new ones coming in July, August, September, and October. So thank you again, and I hope to see you back.
I just have one last comment, if that's possible.
Yes, please.
I just saw questions before that was on the board, but I didn't answer. It'd be very quick. And again, the subject of genetically engineered plant came up again and again. And the question was, what's the impact or the effect of genetically engineered on human consumption and the environment? I'll give a very, very quick answer to that. I highly recommend a report that was done by the National Academy of Science, published in 2016, that put together more than 100 experts in the area of medicine, environment, agriculture, social sciences, and I'll go directly to the conclusion. It shows that genetically engineering plants do not have, to this date, any negative effect on human health, more than other plants. I mean, you can generate a genetically engineered plant with an allergen that can kill you, but why would you do that? So it's same like going and eating a natural plant with an allergen. So they don't do more than harm than others. So that's on human health. On the environment, they do not do more harm than other plants here too. So, and if they are misused, if a farmer is using genetically engineered plants over and over and over and using herbicide, this is an example, over and over, you could see some weeds that develop that becomes resistance to plant. But you could do the same thing with conventional agriculture. So it's not unique to genetically engineered. So the bottom line is I highly recommend this report, and that will give you the answer. And you can't have a better summary than this report on genetically engineered. Genetically engineered plants have been used in North America for more than 25 years, and no single effect, negative effect on human health have been documented or observed. Does it mean it's going to be the case all the time? I would say it's unlikely not, but only the future can tell. That was my comment. I would like to thank you, Professor Aziz Yassir, for this opportunity, and thank ESAFE, PM6P, OCP-Africa, and RUFORUM. It was a pleasure, and thank all the participants for their pertinent questions. I hope I answered them, and looking forward to another opportunity. Goodbye.
Thank you, Professor Fawzi, again, for having clarified the last question. I'm sorry I couldn't give it to you because for the sake of time, but it's good that you have elaborated on. That's thank you. So again, so this is a continuous webinar series, and we are always more than welcome to attend this webinar. So our next speaker is from APNI. So Professor Thomas Obertur. So he will speak about in perennial zone. So you are more than welcome for this webinar, and for all the other webinars that will to inform them possibility to join UM6P to one of the master degrees that we are developing. It is the master degree of fertilizer science and technology. By the way, this master degree has been initiated by Professor FAOUZI BEKKAOUI when he was at school, and it has been initiated in 2018. And thanks for the work he has done for this. We're continuing with this master degree, which is really unique in the world, and it gives opportunities for continuing in research, in research laboratories through PhD study programs, or to join the socio-economic domain in fertilizer industry, in agriculture domain also. So if you would like to apply for this master degree, please send your request to esaf@um6p.ma, or you can discuss with one of our coordinators of this master degree, Mr. Ismail Ghorban, using this WhatsApp Moroccan phone number. Okay, thank you again, and I'm reminding you that you can still watch the whole webinar and the presentation by Professor FAOUZI BEKKAOUI. You have just to go to the ESAFE channel in YouTube, and you can watch it again, and even the question and answer session was recorded for you. Thanks a lot for your participation and for being here and for interacting with the speaker. See you again next Monday, June 22nd, for our next speaker. Bye-bye.